Today we are going to focus on ETC, which occurs in mitochondria. So ETC basically has these functions. It helps in generation of ATP. It helps to create proton gradient, which again helps in flagellar movement in prokaryotes, heat generation and calcium transport. So ETC occurs mainly in mitochondria, which has a matrix, outer membrane, this is the inner membrane space, and this is the inner membrane. If we focus on the inner membrane, the ETC mainly lies over here. So if we zoom in, we can see all these complexes embedded in the membrane. Now first we are coming to complex number 1, which is also called NADH CoQ reductase, complex 2, which is succinate CoQ reductase, complex 3, which is CoQH2 cytochrome C reductase, and complex 4, which is also called cytochrome C oxidase. There is another complex named ATP synthase, which is also called complex 5 or reverse coupling device, because it synthesizes ATP as well as hydrolyzes ATP to ADP and PI. So coming to complex number 1, the NADH, which is produced in glycolysis and TCA cycle, enters through complex 1 and it is transferred. The electrons are basically transferred from NADH to FMN, again to various iron sulfur clusters, which transfers the electron to coenzyme Q. Now in the whole electron transport chain, coenzyme Q and cytochrome C, these two are mobile electron carriers. Other complexes are fixed in the membrane. These mobile electron carriers only moves. So again focusing on complex 1, while transferring the electron from complex 1 to 3, it pumps 4H plus ion from the matrix to the inner mitochondrial space. Then from CoQ, the electrons then flow to complex number 3. It does not associate with complex number 2 when we are considering the fate of NADH. So after association with complex 3, it transfers this electron